There we go. The webinar today will be focused on the sort of recently announced Threatened Species Initiative, which is all focused around empowering conservation action using genomics resources. Dr. Carolyn Hogg from the University...
Doesn't want to stop, doesn't want to do anything.
The University of Sydney, I might just mute a few people here, will be doing the presentation on the initiative today. Carolyn is one of the co-leads for the Threatened Species Project. And perhaps just by way of background, Carolyn, I might just get you to go over to the next slide. Thank you. As a bit of background for those that are not aware of Bioplatforms Australia, I'm the General Manager of the Science Program at Bioplatforms Australia. We are an NCRIS-enabled national provider of research infrastructure in the fields of genomics, proteomics, metabolomics, and bioinformatics, so a number of omics. And we kind of operate across three key pillars. First and foremost, we support a number of facilities, around 16 facilities across Australia, with infrastructure and expertise in those fields of genomics, proteomics, and metabolomics. We also support a national bioinformatics capability through programs such as the Australian Biocommons Initiative. And thirdly, we seed the development of a number of large-scale data resources to help support research themes across things like agriculture and primary industries, medicine, as well as the environment. And these sort of three key pillars enable us to partner with and support a number of diverse research programs across Australia, as well as internationally. Next slide, please, Carolyn. Thank you. So over the last few years, within our environmental theme, we've established a number of national collaborative programs, and these are kind of most of them. And I guess these initiatives have helped to develop a bit of a blueprint for successfully delivering large-scale national initiatives to create these foundational, usually genomics data assets in the form most commonly in things like reference genomes, phylogenomics resources, as well as taxonomy and conservation resources. And together, all of these programs have laid the foundations for our most recently established program, which is the Threatened Species Initiative. And I'll pass over to Carolyn in a moment to dive a bit deeper into what this initiative is aiming to achieve and how you can be involved. But I guess what's probably most important to note here that all of the past and current initiatives are part of a much broader program in this sort of environmental space where we're aiming to, and these are kind of designed to support a large national effort to kind of help build an understanding of the diversity and evolution of life through a foundational genetic assessment of our pretty unique and diverse flora and fauna here in Australia. And TSI is and will continue to work very closely with a number of these projects, particularly those that are still active, such as the genomics of Australian plants, the Australian amphibian and reptile genomics project, as well as Oz mammals and a few other projects we've got coming up in the future. If you have any questions on any of these projects, please reach out to me at any time. But otherwise, I'll hand on over to you, Carolyn, to talk us through TSI.
Thanks, Sarah. Thanks a lot for that. If anyone's just joined, if you wouldn't mind just putting yourselves on mute, just so we can stop the feedback, that'd be appreciated. So before I jump in and start talking about genomes and the Genome Initiative and what we're trying to do with the Threatened Species Initiative, I thought I would just quickly do a summary for those people who are on the call who are not as quite familiar with genetics and genomics as some others. We've got quite a mix. I'm looking at who's on the call today, everyone from some of the country's best geneticists to a lot of people who work in conservation management. So genetics really is the study of specific regions of the genome, and this can be both functional genes, so the genes that do things, or just neutral parts of the genome. And many people who've worked in this space or worked with geneticists for a long time are familiar with microsatellites, which are short tandem repeats within the genome. And more recently, we've moved to what we call single nucleotide polymorphisms, or SNPs, which just can be located across the genome. Where genomics, the study of genomics is really the study of the entire genome. So in the case of mammals, it's about a 3 gigabase genome, or it could be 3 billion base pairs. Now, for the non-specialists or non-geneticists in the world, geneticists have a really bad habit of interchangeably using the term genetics and genomics. And so it just adds to the confusion. We like to keep everyone on their toes. So how do we simply study a genome? Well, we like to get a sample to collect, preferably not a liver from an animal, but for some of the species that we do work on, we do like to get fresh organs, and that's because we need transcriptome data in order to annotate the genomes. But really what happens is we get the sample, extract the DNA, we do a library prep, send it off for sequencing. It gets sequenced at the sequencing company, and then we get these large data files back. So we get significant amounts of information back. But essentially, and we do this both with a whole genome as well as the SNP markers as well. So SNP markers are very much like just getting different pieces of a jigsaw puzzle. So if I was to give you the 5,000 piece jelly bean jigsaw puzzle, of which I only give you 1,000 pieces, and I ask you to put them on the table in the right place, it's quite hard to do because you're not quite sure where the piece goes. However, if we give you the puzzle box lid, then it makes it much easier to start to understand what the pieces of the jigsaw puzzle, where they sit on the table, and also what their function is. And so the puzzle box lid is really the reference genome. So when we talk about creating reference genomes for species, we're trying to create a series of puzzle box lids. So when conservation managers or other people go and get their DNA sequence, they can understand what the pieces of the puzzle mean. So if we look now, this photo, Earthrise, was actually taken by Bill Anders back in 1968, almost 52 years ago on the 24th of December, and during the Apollo 8 mission. And this photo has actually been declared as one of the most influential environmental photographs ever taken. And I like to use it in my talks because I think, for me, it's one of technological marvel, and it tells a story of fragility and of hope and of biodiversity. And the interesting thing is that when Bill Anders took this photo, the internet, artificial intelligence, machine learning, genome sequencing, smartphones, even the lowly Walkman were all actually things of science fiction, because none of those things have been created 52 years ago. And you think about now, all those things now exist, and the rate of technology is simply accelerating. Unfortunately, though, in that same 50-year period, over 330 animal species have gone extinct, with the rate of extinction simply accelerating. And these days, you don't need to go far, whether it be in the public literature, in the media, in the grey literature, as well as scientific peer-reviewed publications. Everywhere, everyone is talking about this biodiversity crisis that we find ourselves in. And then if we look at the IUCN Red List, of the 13,500 animal species that are listed as conservation dependent or worse on the Red List, less than 0.8% of them have a genome. So we have all these reference genomes being created at a global level, but not many of them are actually being created on threatened species. And the reason for that is vast and varied, but one of the predominant reasons is actually very difficult to get samples of threatened species. If we look at the global genome repository database, there's 712 plant species. And I'm really sorry to the plant people on the call, they only break it down to green algae, land plants, and other. So anyone who works in plant genomes, you might like to get onto NCBI to make their categorization easier to see. And then there's actually, in fact, over 2,500 animal genomes. Most of those genomes are, in fact, invertebrates and fish, followed by, of course, by mammals and birds, and then the other taxa. And if we look at the 2,500 animal genomes that are on NCBI, only about 6% of those published genomes belong to conservation-dependent species. So although there's been lots of commentary in the literature about the use of genomes for conservation, the resources for threatened species in this space are still quite limited at a global level, but it is accelerating quite quickly. And the reason for this is because the cost of genome sequencing has come down dramatically since 2001, when the human genome was first getting done, to it only costs probably about $10,000 in Australia now to do a reference genome for an average size genome. And we see this as well in the amount of genomic sequence data that's getting published. So the blue line is just the normal gene sequence data that's being published over time, but the red line is very much the whole genome data that is getting publicly released. And as you can see, as the cost of genome sequencing is coming down, we're getting this exponential increase in the numbers of whole genome sequences, and also a very large expansion in what we call big data sets. So the compute power we now require to be able to analyze all this data is becoming more problematic. And so this leads us to what I like to call the Great Divide. So in the Great Divide, you find on one side, we have the geneticists and genome biologists building this amazing genomic library and this amazing resource. However, on the other side are the ecologists and the conservation practitioners and the policymakers who are starting to tear their hair out, because every time you get your head around what a microsatellite is and what it does, we change the technology and we've moved to SNPs. And once you just start to understand what a SNP is, we've changed the technology again, and now we've moved into whole genome sequencing. And every time you get your head around what whole genome sequencing technology is being used, it's getting changed. And the speed at which the technology is evolving is becoming more and more rapid. And so what's simply happening is this divide is actually getting wider and wider. But one thing we do know is that if we can bridge this gap between what the geneticists and genome biologists are doing and what the ecologists and conservation practitioners and policymakers need, we can hopefully start to contribute to ways in which we can lower some of our extinction rates. And it's not because managers don't know genetic diversity is important. I haven't met a conservation manager yet who doesn't know it's important for their system. They know that diversity and inbreeding are inversely related, that it's important to maintain adaptive potential for future changing events and for individual fitness to ensure survival of species. But here in Australia, genomes in conservation have always traditionally just been considered as a nice to have. And so for my multitude of sins, I sit on a number of recovery teams. And in these recovery teams, when we talk about using genetic data to make decisions for management purposes, it's commonly commented on that we just don't have the resources. As we all know, resources for conservation application is pretty limited. And so you have to really choose where you're going to invest your resources. However, when I spent a very lonely Sunday afternoon late last year, looking at all the national recovery plans. So of the 200 vertebrate recovery plans we have in Australia, more than 80% of them actually list a genetic action in the recovery plan as a way of helping the recovery of the species. But less than 15% of them have access to or are using genetic data to help inform those decision makings. And so really, if we have genomics in management, what does that mean and what will it actually do? Well, once you've got a reference genome of your species of interest or a cogeneric species, you can then characterize functional genes. So people who are interested in disease, we can look at the immune genes in relation to the disease that the animals are suffering. And 3 of the species you can see on the screen here are 3 of the species we work in our group in relation to disease. We can identify the reproductive genes, behavior genes, heat tolerance genes, which are associated with how animals are able to adapt to the different climatic environments that we have here in Australia. Hopefully will help be more informative moving forward into the future in relation to climate change. And then of course, we also have our genome-wide diversity data. So this allows us to differentiate between populations, look at the genetic diversity within and between populations, as well as inbreeding effects. And the list really goes on in the types of questions that you can ask based on genetic data. And so when we went to a series of different management questions that can be asked and answered, and there's a lot of information on this slide and you don't really need to take it all in because all these questions are actually listed in our communique on our website. But it's just to give you an idea of the depth and breadth of the types of questions that can be asked and answered if you have really solid genomic resources. And so what you need to think about with genomes and genomic resources is they're just a tool. And they're not the end point, but that they are the starting point that you can use to ask a suite of different ecologically based questions. And for us here in Australia, of course, this is becoming more important, particularly after the horrendous summer that we had last year, where You know, we lost over 1 billion animals were lost in the bushfires, with 20 million acres of habitat burnt in the environment. And here in Australia, we have over 8,000 vertebrates and more than 24,000 plant species, with most of them actually being endemic to the country. And a recent study that just came out in Nature Ecology and Evolution looked at what kind of faunal habitat was impacted by the fires last summer. And as you can see, a large portion of their habitats were burnt, and for some species, they lost significant portions of their habitat. And so that's where, you know, management actions have been taken, whether it's go in and bring the animals into captivity for the short term while the habitat regenerates. Do we need to translocate the species from one site to another to help repopulate those sites because they now live in a very fragmented landscape? Or do we bring some of those animals in and start up a breeding population? So for all those different kinds of decisions around wildlife, and it goes for plants as well, having genetic resources will really help inform some of those decision makings. And so this is where the Threatened Species Initiative really comes into fore. So unfortunately, we're a bit late for what happened with the bushfires last summer, but if we build the genomic resource now, it'll be a capacity that we can use and a resource we can use in future catastrophic events moving forward. So the plan is, it's a joint initiative. So myself at the University of Sydney, Kim Ottewell at the West Australian Department of Biodiversity, Conservation and Attractions, and Peter Latch from the federal government, and ourselves joined together with Bioplatforms Australia to start the Threatened Species Initiative. So the three of us are the co-leads for the project. And the idea is to create genomic resources for some of Australia's most threatened species. That's both animals and plants. And we're hoping to do somewhere between 40 to 50 species, depending on what the needs and the requirements are. The other thing we'd also like to do during this project is not to do a reference genome for every species, but try and do reference genomes for a representative species of a family, and then provide population genetic data for multiple species within that family, and they can align to the one or two reference genomes that we create. So we also are going to fund associated population data. Now, this is not to do population data for every individual in a population, but really it's to fund 1 or 2 sequencing plates for a population as a kind of a launch pad or a starting point. So this project is not about creating a resource, lots and lots of resources for a very limited number of species. What we would like to do is to create a large number, a small number of resources for a large number of species. So think of it as a launch pad or a place that you can start, get some funding and support to get your genomic data started, and then you can go on through the process. And the last but not least is one of the things that we're really keen to develop during this project is an applied conservation genomics portal. And this really is an online toolkit mainly for conservation managers or research scientists that are working outside of the academic sector, or for ecologists or any non-genetics specialists. And the toolkit is really designed to help people who want to use genetic information to use it without having to become an expert geneticist themselves. So the Threatened Species Initiative really is about reference genomes being assembled and annotated, not only by the TSI, but the other, as Sarah introduced, the other bioplatforms initiatives have created a suite of different genomes. There's also a number of global initiatives which are creating genomes, and these are all publicly available in the cloud, whether it be on NCBI, bioplatforms, or with Amazon Web Services. But the way the concept is meant to work is that if you're an end user, you go out in the field, you collect your sample, you can send it off to a commercial provider for extraction and sequencing. You get your data back, very exciting moment. It's lots of little letters or numbers on a screen. What do you do with that information? And so the plan is eventually that you'll get your data back from the commercial sequencing company for your populations, and you can load it into our cloud-based portal. So I must thank Paris Brandes from in our group, who is the architect behind the mock-up that you're about to see for our cloud-based portal. So the idea is that you'll be able to come to the portal and you can look at the tutorial. It will be highly advisable to do the tutorial the first time you ever use the portal, and then you can get started with your data. And so you can come in and you can say, well, I want to align my data to a reference genome, or I just want to assemble it because there's no close reference genome at the point of time. And if you hover over things, you'll get little pop-up windows to provide you with information. You can then select whether it's from a TSI database, or maybe it's already, there's a genome that's already been created and it's loaded up on the international repositories. You then input your data files that you receive from the sequencing company. And then you check the boxes off for the downstream analysis. So whether you're interested in genetic diversity analysis initially, or maybe you wanted to do some parentage analysis for your population. There's some advanced parameter settings which you can be able to adjust, but there will be a series of default settings. And then you put in your submission details, hit submit, and you get a submission summary. Now, what happens in the back end once you've done this is it actually fires up a computer in the cloud. It runs all the analysis and it gives you a standardized report. So this is the idea. It's probably going to be a pay-for-use service unless we can find an organisation that's willing to sponsor it at a really high level to help the ongoing maintenance costs. But the idea for the pay for service is really to pay for the commercial cloud computing. And the reason we've opted to go for commercial cloud compute rather than one of the free services at this point in time is so those of you who don't work in the academic sector or the research sector, like conservation managers or people who work for wildlife agencies or zoos, can actually still access the portal as well, because you don't have an academic, if you don't have an academic email address, it can be difficult sometimes to access cloud computing. So once you get your data back, it will come in an easy to interpret format. And then you can simply use that information and apply it for your management decisions, whether it be translocation or captive breeding, habitat protection, bringing plants or animals into a seed bank or to a zoo, any decision, and go around again. So to give you an idea of how fast we can do this, lots of these photos have myself or our team members in it. This is what we do with the DEVIL program at the moment. We collect the samples in January, we sequence them, we process the data, and in June, we're out in the field making the selections for the translocations and the harvest. And so we can actually do this process in a period of about 4 or 5 months, but that's once you get it up and running. So where did TSI come from? TSI really started with a conversation that I had with Peter Latch around about 2018 about identifying the need that genetics and genomics is really important for conservation management, and how can we better assist conservation sector in using this information moving forward. And so we sought a partnership with Bioplatforms Australia, and then spent 2019 doing some initial consulting with the Nest Threatened Species Hub, the threatened species managers from all the states and the federal government, as well as more in-depth detail with New South Wales, Queensland and Tasmania, and some of the NGOs around the country. And then we were planning on having a series of workshops starting in March 2020, but we all know what happened. So COVID happened, and we didn't do those workshops. But that didn't stop us from deciding to launch a major project in the middle of a global pandemic. So we launched the project in May this year, and we started the pilot phase in August. And so at the moment, the steering committee consists of these 8 people. Maurizio is located at the Royal Botanical Gardens here in New South Wales, and he's a geneticist who works a lot with plants. Kathy Belov's from the University of Sydney. James Biggs is from the Zoo and Aquarium Association. Kim I've already talked about in Western Australia, and Sarah and Andrew from Bioplatforms Australia. So the steering committee at the moment is really just trying to build the framework and build the way that the project is going to evolve over time. So we've already commenced a pilot project, and with the pilot project, what we wanted to do was ask and answer a number of different questions in relation to the workflows that we're creating. So we wanted to make sure that once we go out for the larger call for partnership and people start becoming involved in the initiative, that we had kind of tested all the workflows to start with. And so as you can see, there's a number of different scenarios that we have in regards to the pilot project, and I'm more than happy to talk to anyone later on about what these different things mean. But what we've done is we've started with the eastern bristlebird, the swan galaxid and native guava. These are all species that have no reference genome or no genetic data at all, and you can see a lot of them have already been sequenced or awaiting sequencing. We also wanted to look at what happens when you have a closely related reference genome. So the Masticomys genome is already published, but there's no genetic data for the Hastings River mouse. So that's being done at the moment. We have lots and lots of population genetic data for the orange-bellied parrot, but we had no reference genome, but the data arrived back yesterday, which is great. And then we have a reference genome for the eastern barred bandicoot, but the eastern barred bandicoots and western barred bandicoots, or Shark Bay bandicoots as they're now called, have some very distinct populations, and a lot of the management decisions for these two species are around mixing these populations. And so we wanted to see whether or not we could use one reference genome from one species and make it informative for another species. And then, of course, we have the ghost bat, which has no reference genome and has some genomic data and some population genetic data, but it's not really a full data set. So we wanted to see what it would take and what the associated costs were for these different scenarios. So that will help inform how many other species we'll be able to do throughout the course of the project. So part of the reason you're here attending this webinar today is because we've started the request for partnership. It's located on our website. You can go to the website and find the updates, and if you click there, you'll be able to find the full list of species. So what we've done is, rather than asking people to just nominate the species that they want to work on, we went out to the threatened species managers across the country at both the states and the federal level, and we asked them to prioritise their top 10 species for their states, and only in the animal space. And the reason for that is because plants are a lot more complicated. So what we've done is we have now got a list of 57 species across Australia, and those are the different taxa. For those of you who want to work in plants, please nominate your plant that you want to work on. And the reason for that is because we're trying to work very closely with the genomics plants team to make sure that we're not doing cross purposes. And with plants, my understanding with plants is that it's more around some of the conservation actions and the need for those multiple plant species that occur in an area than specific species. And so the advice we received when we were doing the initial consultation process is that we shouldn't provide a list for plants, that we should ask people to nominate a plant. For those of you who are on the call that actually sent us your list of species, when you go onto the species list in the Google Doc, you'll see that there's two tabs. There's one tab which is the priority list, and those are the 57 species that have no genomic data or genetic data for them, and we need to create it. There were, however, about 33 species that were nominated from different state and territory agencies that we know that there's either a genome or there's some genetic data in existence and there's people working on it. And so rather than simply leaving those out there for you to try and figure out who those people are, if you contact Sarah, we'll put those conservation managers in touch with those academics or those researchers who are working on those species to help facilitate the partnership at the moment. So the reason we did the request for partnership, we advertised it now. The idea is that it will give academics and researchers who want to work on different species the opportunity to contact the respective state agencies, or if they're not sure who to contact, we can contact us, or contact Sarah, I should say, and we can put you in touch with the right people to try and facilitate that discussion around who you should be speaking to for the partnership. Because the criteria for the partnership is really around end user collaboration. So part of this project is to ensure that we can put the right people who are working on the genetic data in the academic world or the research sector in touch and in connection with the people who are going to use that information and develop a collaboration. And so we're going to open the formal call for partnerships in early February, and we'll make the announcement in March, which means if there's anyone who would like to put in an ARC linkage project or start developing an ARC linkage project, those submissions are not due to probably April next year, and then the next round is in June, July. So it's just to help with the co-funding part of the criteria. So the species must have an active management plan or a conservation plan, and the plan must identify or be informed by genetic information. So you can't just pick a species that you like but has no conservation action. We're trying to facilitate in the first instance management around species that, if they had the information in the next 12 to 24 months, how informative would that be for them to make decisions moving forward? Also, you need to have capacity, bioinformatics capacity to do the genome data or the companion data sets. We do have a bit of a bioinformatics network within the Bioplatforms Australia initiatives. So please, if this is a massive area or a gap for you, please do contact Sarah, and hopefully we can put you in touch with some right people. Scientific rigor, the samples must be available or easily obtainable. The biggest crux or biggest sticking point, two biggest sticking points for genomic research is one, being able to get the samples to do the research, and two is the compute required to compute the large amounts of data. So hopefully we fixed the compute part of the equation, but the samples being available is really left to those guys who are on the ground. And then, of course, there needs to be a co-contribution of funding to complement the BPA investment. So the bioplatforms investment is very much around paying for the sequencing, and that's to sequence the reference genome and also the population genetic data. And then you need co-investment around how you're going to analyse that information and apply it, and also how you're going to collect the samples and extract them. So we've just started developing some working groups. Chris Helgen has generously offered to chair the archiving and collections working group. And what the plan is that any of the samples that are collected for the threatened species project, particularly the genome samples, they need to be permanently archived in a collection, so that not only do we end up with the reference genome, but we also know where the organism, whether it be an animal or a plant, that created that genome is actually stored. So Chris and the team are currently creating a series of protocols around how to do those sample storage. Conservation reporting, Kim Ottewell is leading a group at the moment that's got myself and Paul Sunnix, Andrew Weeks, and a few other conservation geneticists around the country. And we're trying to come up with some standardized metrics that we think should go in the reporting. And once we've created that, we'll expand that group to bring in some conservation managers to give us feedback. So this is for the portal portion of the project where you receive that standardized report. And what we want to do is see if we can standardize those reports across the country, so people or the end users who are using that information get the same consistent information over time. Rich Edwards is looking after the bioinformatics scope. He also sits on the ASAG team, so he's the best placed for bioinformatics. And Sasha Pavlova is an excellent population geneticist down at Monash in Paul Sunnix's team. He's going to help us with the population genetics component. So at the moment, the timing for the project really is, as we've said, we've released the phase 2 species list. We're going to do the formal request for partnerships in February 2021, and we'll make an announcement in early March 2021. And then part of the project is to revisit the current list with the threatened species managers later on next year. And this is just to account in case there's some other significant catastrophic event that happens in 2021, and I think 2020 has taught us all that catastrophic events come pretty fast and furiously at us. And we just wanted to keep a little pocket of money aside in case something happens next year, and we can release another phase 3 list roughly the same time next year for 2022. And that the development of the online portal, the large development will happen in 2021, early 2022, and there's a few people who have already put their hands up to be beta testers. We're hoping to have the online portal fully up for people to use by mid-2022, but as you can imagine, there's still quite a lot of development that needs to go around that. So just to give you an idea, these are some of the organisations that we've been talking to in regards to the Threatened Species Initiative over the last few years, and we've had really positive feedback and a large amount of support from people who think the project has value and merit. I particularly want to just flag for you AWS Pawsey Supercomputer in Western Australia, the Ronin team and Intel, and the Australian Biocommons. These are all the compute folks who are really trying to help us figure out ways that we can facilitate the rapid analysis of some of these very large data sets, so you're not waiting months and months and months for the data to come back. So at the end of the day, I started this talk really with this very large, wide gap, which is actually getting wider with the way the technology is working. But we like to think that here at the Threatened Species Initiative is that really we're going to build this bridge and hopefully create something that will probably have quite a few hiccups along the way, but by the end of the project, we'll have created a really good foundation and a good launch pad for using genomes in conservation management here in Australia for both fauna and flora. So if we come back to where I started, this photo was taken over 50 years ago, and the technological advances are simply excelling, but the rate of extinction is also increasing. And so what we do know is that genomic resources for on-ground conservation action are highly useful and can be used for a wide range of applications. And we've seen that in some of the most iconic species we've been using it for in our research group. Of course, that's the Tasmanian devils and more recently the bilbies. And so I just wanted to leave people to ponder the questions of, for those of you who are on this call who are creating genomes, how are we making them useful for conservation managers, and how do we better articulate the value and use of the science that we're creating? And really, how can we use the resources that we're going to build through TSI, and this is both for the managers who are going to use the information and the people who are going to create the resources. How are we going to join those two forces together to help our actions really have impact? Because more importantly, what is this planet going to look like 50 years from now? So that's the end, and take some questions. I think they've been coming in on the chat, and if you want to just reuse the raise your hand function, and Sarah, I believe, is going to facilitate this part of the call.
Thanks very much, Carolyn. Very powerful ending to that presentation. Let me just check. I don't think I've seen anything come through on the chat. So perhaps if anyone has any questions, yeah, feel free to use the raise your hand function, questions or comments. And feel free to put your video on just so it makes it a bit easier to chat. You've stunned everyone into silence, Carolyn. Oh, here we go.
I've scared everyone.
All right, we've got a question come through from Shandia. Is there a timeline for when the genomics portal will be available to end users?
Shandia, if I had my way, I'd be ready now. It's interesting, I gave this presentation at the Global Biodiversity Genomics Conference and I got the same question, but everyone around the world wants it. The hope is as quickly as we can. So we have all the components. Arthur Georges has created the fantastic Dart R package, and we have some other custom scripts that we've built. And the framework for the platform is there. We now really are just trying to seek resources at the moment to fund a software developer in 2021 to actually build the actual infrastructure platform itself. So I'm hoping that there'll be a beta version by the start of 2022. And if you want to try and break it for us, Shandia, that would be useful.
Any others? Mike Gardner, will the portal be free to use for species where resources have been generated into TSI?
I would like to think so, Mike. At the moment, I'm just trying to finalise some cloud resources for the TSI project from Amazon Web Services, which means it will give us free cloud compute to use the portal for the TSI species. And definitely we're looking very positive until COVID hit. Unfortunately, a lot of what was going to get allocated to us got taken away for medicine this year. And so we're just building a business case at the moment to try and get that back. But the hope and the plan is to provide it free of charge for all the TSI species.
Yeah, I think also beyond that for the portal, the aim is that for any compute that does need to happen, there would be some kind of estimator to give you an idea of price. And I know that Carolyn's team have been doing lots of these different pipelines and already have pretty good ideas of how much these things cost. And it's not like the compute time, and it's not huge. It obviously is species dependent, but can be as little as what were you telling me the other day, Carolyn? $20 US to
it's $26 US to assemble a hi-fi genome.
Yeah,
in the cloud. So it depends on the compute requirements, but yeah, anyone who's interested in the associated costs with compute and the different analysis pipelines, just send me an email and we'll be able to provide you some information back.
We've just had a question come through from Gil Brown. Is there any chance for coordinating a plant prioritisation group through TSI like you had for animals, or is this something that should be discussed through GAP?
I'm going to leave you to answer that one, Sarah, because I went on holiday and left Sarah to have the plant call. So, yeah.
Yeah, so Gil, we have been in very close contact with GAP as one of our other framework initiatives, and are kind of consistently talking about how we might sort of do that prioritisation. I think we'd be very open to extending that and having a discussion. What kind of came out of that was that it's a lot more extensive and complicated for plants, as sort of Carolyn mentioned before. So there was no kind of quick or really effective way to do that in the time frame that we were looking for to kind of launch this first round. But certainly as the community nominate plant species to be sequenced as part of the Threatened Species Initiative, we'd love to have a group that could help us evaluate those based on their conservation importance, because that can be very tricky. So that was sort of the driving force behind wanting a list for animals, and I think that holds true for plants. It was just a bit harder to logistically manage. So if you have any ideas for that, please send me an email, and we'd be happy to discuss how we could potentially do that. Another question from Claire. Particularly love the idea of standardised reporting. I did wonder what your thoughts were regarding interpretation of data for conservation programs, if this is a gap that is closing.
Well, the fact that we've got about 6 conservation geneticists on the working group at the moment who can't agree is probably an indication of currently where we sit. As anyone knows, interpreting genetic data can be quite fickle, and a lot of it has to do as well with how you filter your data sets, how you enter the data, how many populations, your sample size. There's a number of different factors. And so what we're trying to do is come up with a way of capturing all that within the reporting. So I think what we've agreed to is, with the reporting, we're going to start at the easy, low-hanging fruit first. You know, what is heterozygosity, what is the inbreeding coefficient, those types of statistics, and then move into the more complex conservation management. So I don't foresee us in TSI, it's not easy by any means, but it is achievable, but it is going to take quite a bit of work. And there's a global program at the moment also trying to come up with these solutions. So the idea is that through the Threatened Species Initiative, hopefully we will build and develop something that becomes more useful for conservation managers. And it's also about trying to connect conservation geneticists with people on the ground who are using the data to help accelerate that process of learning and tuition. And the plan is, probably starting in mid next year, is start to have more regular webinars where, you know, we can do some really basic training in how to interpret and understand the data.
We have another question from someone who will ask it live, but for some reason your name has come up with about 50 letters in the chat. So I might just open the floor for whoever.
Hi. Yeah, it's me. Sorry, I don't know why that happens. David Field from ECU. Yeah, I have 2 questions, if that's allowed. And great talk, Caroline. I really enjoyed it. It hit the mark with me. The first one sort of leads on from the previous question. Yeah, I think it's a great idea to have standardized analysis for genomic data, but as you put forward, it's actually very complicated, and I think there's a lot of scope for gross misinterpretation, incorrect analyses that could have, you know, incorrect application for managers. So I just wonder, it will be very difficult, I think, to automate such a thing, and you really need geneticists and theoreticians involved to probably to filter every single analysis output and interpretation of that. It's not
standardizing the analysis. The analysis needs to be different for every species, and that's a given. It's standardizing the reporting metrics. So when you give the report to a conservation manager, the reports look the same and the content of the report is the same. And I can say this because when you read a multitude of genetics papers on the same species, they all use different analysis. Some don't report heterozygosity, others report it different ways, inbreeding is reported differently, some people don't put in sample sizes, some people don't put in standard errors. all those kind of things are the things we want to standardize. And so the hope is that over time, as conservation managers start to become more comfortable with the information, they know what they're looking at. But yes, we fully agree that the how to develop those metrics will need to be individualized, and we'll always need geneticists to help. But part of the problem we also have is, you know, there's a large range of people who did a bit of genetics during their degree and now they're an ecologist, and they want to just get some data back from DART, and they want to run an analysis, not because they want to report on the genetics of their species, but they're really interested in whether or not they've got a reproductive skew in their population by a certain male. And that's just a parentage analysis. So it's trying to help provide a place where people can go and get that information and the tools and that type of scenario. And part of that is what the Australian Biocommons is trying to do, but you still need to be quite an expert in understanding genetics. So we're trying to just fill the gap between those two projects at the moment. But yeah, I agree with you. Standardized filtering is never going to work. Although we'd be so lovely if it did.
Yeah, yeah.
Do you have
you had two questions.
Yeah, I had another one. So yeah, another thing, as you put forward, a lot of adaptive traits for species, you know, are most likely going to be polygenic in nature. And so some projects will also be collecting phenotypic data. Is there any scope for collecting quantitative phenotypic data on individual population level data?
I would like to think that the people who are coming to TSI to obtain the genetic data sets are collecting phenotypic data. We do have quite a comprehensive metadata that needs to come in with all the samples, but it's not that kind of comprehensive phenotypic type data. But I think that's a really valuable, and that's particularly for plants, I would say, and to some extent some animal species. But yeah, we'd be really open to having that discussion about how do we better facilitate that information to be joined with the genetic data. Maybe in the BPA portal, Sarah, is a place we can link the data sets. One of the things, we actually put in a funding bid to the ARDC, but were unsuccessful. And we were proposing on building a massive genomics online hub, which would talk to the ALA and do all sorts of fancy things, which I think would still be really useful in the long term. But in the short term, we'll just do the short genetics analysis bit now.
Thanks.
Thanks, David. Any other questions? We've still got a couple of minutes, so feel free to ask away. All right, we might call it there. But in closing, thanks again very much, Carolyn. That was a fantastic talk and a really good insight into what TSI is hoping to achieve. Anyone and everyone who's interested in being involved, please feel free to reach out if you have any further questions. My email address is on the TSI website, and you feel free to contact me with any questions about TSI or any of our other bioplatforms initiatives. And yeah, we look forward to hopefully working with many of you as this project begins to really progress over the coming year or two.
Great.
Thanks everyone for your time.
See you later.